Despite decades of reliance on animal models, poor clinical translation remains a major challenge in drug development. In oncology alone, more than 90% of candidates that show efficacy in animals ultimately fail in human trials. To address this gap, regulatory agencies and the pharmaceutical industry are advancing new approach methodologies, or NAMs, with the goal of improving human relevance while reducing animal use. NAMs refer to a suite of methods that generate data in lieu of animal testing, following the 3 R's: replacement, reduction, and refinement. In-chemical methods are chemical methods that test a substance's reactivity at a molecular level without using living cells or organisms. In silico methods are computer-based tools using AI, math, and big data to simulate and predict biological effects. In vitro models include spheroids, organoids, and more complex microphysiological systems like organs on chips. These are human-relevant, non-animal testing methods to assess the safety and effects of chemicals and drugs. Among in vitro NAMs, advanced human liver models are playing an increasingly important role in improving translational relevance and helping to reduce drug attrition rates. In vitro liver models are central to studying liver function and predicting human responses. Traditional 2D hepatocyte cultures are simple, well-established, and useful for short-term studies. However, they rapidly lose liver-specific function, including drug metabolism and enzyme activity, which limits their ability to predict human outcomes. In contrast, 3D hepatocyte spheroids are clusters of primary human liver cells cultured in 3 dimensions. They self-assemble into structures that closely mimic the architecture and function of human liver tissue, preserving cell-to-cell interactions and have tissue-like architecture. They maintain metabolic and functional competence over longer periods. This allows for more human-relevant studies, not only of drug metabolism and liver-mediated toxicity, but also for broader mechanistic and translational research, including chronic exposure studies and emerging therapeutic modalities, including gene therapies. 3D hepatocyte spheroids can be applied for a wide range of studies. ADME and toxicity-related applications include human-relevant evaluation of drug metabolism, enzyme induction, and assessment of drug-drug interaction liability and hepatotoxicity over extended periods. Beyond the traditional ADME-Tox studies, these models are increasingly used for gene and RNA therapeutic development, for mechanistic disease modeling, and in translational studies. While 3D hepatocyte spheroids offer significant advantages, their success depends on several key factors. For example, the donor-derived liver tissue can impact success. Donor-to-donor variability can impact the ability of hepatocytes to form spheroids, and even the time and culture it takes to form spheroids. Additionally, the type of low adhesion vessel, which is used for high-throughput spheroid formation, can also influence formation and functionality. Human hepatocytes from different donors vary in their ability to self-assemble into 3D spheroids. Not all hepatocyte lots form spheroids despite plating well in 2-dimensional monolayers. Take donor 1, for example. The hepatocytes remain very loosely aggregated and fail to form intact spheroids even by day 14 of culture. And among donor hepatocytes that are capable of forming spheroids, differences in aggregation efficiency can influence how quickly spheroids form in culture and how stable they remain over time. For example, hepatocytes from donor 2 had already self-aggregated by day 8, showing spheroid-like morphology. This is compared to hepatocytes from donor 4, where the hepatocytes still remain very loosely aggregated at day 8. These donor-dependent factors can directly impact downstream functional readouts, making it important to screen donors before using them for 3D in vitro cultures. Even the type of ultra-low adhesion vessel can make a difference. For high-throughput formats, ultra-low adhesion, or ULA plates, are often used for spheroid formation. Here we are looking at data comparing spheroid formation over time in ULA plates from 3 different vendors. You can see on the left, spheroids successfully formed in all 3 plates, facilitate in sphero and alplasia plates. However, on the right, you can see that the speed of spheroid formation varies across plate types. Spheroid formation occurred much faster in sphero and alplasia plates compared to the facilitate plates. Across multiple hepatocyte lots, spheroid formation took on average 9 days in the facilitate plates compared to only about 5 days in the sphero and alplasia plates. As new approach methodologies move preclinical research towards more human-relevant non-animal models, 3D hepatocytes support sustained liver function for ADME and toxicity studies, as well as advanced hepatic research. As we have demonstrated, successful implementation of in vitro 3D hepatocyte models depends on primary hepatocyte quality, donor selection, and technical expertise. As leaders in hepatocyte innovation, Discovery brings deep hepatic science expertise to ADME and emerging applications such as gene therapy. To learn more about our primary human hepatocytes for 3D modeling, email us at info@dls.com.